So many of the presentations preceding this one have all involved neurodegenerative disorders. So it's safe to say that it's a large focus of study and it impacts a wide number of people. So a little bit of background about neurodegenerative disorders. It's a loss of structure or function in neurons, specifically in the brain. And so to name a few, Alzheimer's, Parkinson's, and Huntington's all have this loss of neurons. And so what's been found is that neurotropin levels are reduced in neurodegenerative disorders. So what neurotropins are innately expressed proteins that promote the survival, growth, and regeneration of neurons. So, as you can imagine, patients with these neurodegenerative disorders, in this case Alzheimer's, have a lower concentration of BDNF, a neurotropin. But in patients without neurodegenerative disorders, have a normal concentration of BDNF. So their neurons are being protected by BDNF, this neurotropin. So BDNF falls in a family of 4 of neurotropins, and we focus specifically on BDNF. And one of the reasons why is because BDNF is expressed in the hippocampus. And the hippocampus is largely affected by neurodegenerative disorders, specifically Alzheimer's disease. And so when BDNF binds to TrkB, TrkB is its receptor. It lies in the family of tyrosine kinase, so TrkA, B, and C. TrkB also is expressed in the hippocampus. So BDNF binds to TrkB, which activates autophosphorylation and dimerization. This goes on to activate other pathways, which in turn activates neuronal survival and protection of the neurons. So, although BDNF looks like it's a great neurotropin, recombinant BDNF, we run into some issues for therapeutics. So BDNF has a short half-life, so it's not potent and it's very, very unstable. So it can't get to the parts of the brain that we need it to in the time we need it to. So although it is innately expressed, recombinant is a bit promising for a drug. And also a huge roadblock is it can't pass through the blood-brain barrier. So as you can imagine, this is not promising for a therapeutic in neurodegenerative disorders. So this is why we look for alternatives. So flavonoids are a secondary plant metabolite. So these aren't expressed in mammals, they're found in plants, such as fruits and vegetables. And so what's interesting is that flavonoids have also been shown to mimic BDNF, so it's able to promote survival and protection of neurons. And so flavonoids bind to the TrkB receptor, activates these pathways, and also may cause this neuroprotection. And so 7,8-DHF, a flavonoid, has been shown to imitate BDNF. So here, the top row, you see phosphorylated AKT, which is a pathway that's activated by the ligand binding to the receptor. So BDNF, as you can expect, has phosphorylated AKT. Interestingly, 7,8-DHF also has phosphorylated AKT. And as you see down here, the morphology of the neuron in the instance of 7,8-DHF hasn't been damaged. So 7,8-DHF isn't toxic, which is a huge potential as a therapeutic. So 7,8-DHF has a longer half-life. So it's more potent and it's way, way more stable, which is really promising quality. Also, it's able to pass through the blood-brain barrier, which is also a huge quality that you look for when developing these drugs. So my objective this summer was to test these candidate molecules that are similar to 7,8-DHF and develop assays to kind of test the efficacy of these candidate molecules. So they're similar to 7,8-DHF, but there's a little difference in the structure of them. So one of the first things to test is this interaction between the ligand and the receptor. As I said before, upon the binding of the ligand to the receptor, you have autophosphorylation and dimerization. So I looked and, So I was looking for an antibody to correctly label for phosphorylated TrkB. So what I did is I treated cells with BDNF, and I treated them with BDNF as a positive control, because I know BDNF binds to TrkB receptors, so we should see phosphorylated TrkB. So I treated cells with BDNF for 30 minutes, and then I took the lysates and did a Western blot. And what I found was that although there are bands, they don't lie within where I thought, within the size of phospho-TrkB. So it's about 92 kilodaltons that I would expect to see bands. However, I see bands all across and down below, which should not have been phospho-TrkB at all. And also, cells, TrkB expressing cells with BDNF and without BDNF also saw some labeling. So what I found was that our antibody that we had ordered had non-specific labeling, so it didn't label correctly the phospho-TrkB that I wanted to investigate. So another assay that I developed, or that I attempted to develop, was to look at how this flavonoid binds to the receptor, activates these pathways, that in turn activates or promotes the neuronal survival. So before I could progress and actually test our candidate molecules, I had to do a cell titration. So I needed to see the optimal number of cells. So I needed to see the greatest difference between the cells that were apoptotic or the cells that were not. So what I did was I treated my cells with STS, staphylosporine. It's a kinase inhibitor, and that induced an apoptotic environment. And so what I did is I plated my cells and let them incubate overnight to let them acclimate to their new environment. And then I pre-treated them with my flavonoid derivatives for 30 minutes, and then I pre-treated them, or I incubated them with STS for 9 hours. And so after that, I added a reagent that's not really in the literature yet, so this is also really new and interesting. I added a reagent called cell titer glow from Promega, and this luminescence in the presence of ATP. So cells that are alive or living have higher concentrations of ATP, whereas cells that are dead or dying have lower to no concentration of ATP. So you would imagine in the cells that are alive, luminesce brighter. So what I did is I did a cell titration, and I plated 0 to 50,000 cells. And as I said before, I needed to do this before I could do my further experiments. So what I found is that the greatest difference between the treated and untreated lie about 25,000 cells. So that's what I plated in my future experiments. And so with this number in mind, I was able to go on and test my candidate molecules. And so in the ones that are 100% live, these were not treated with my candidate molecules nor STS. So interestingly, I found that it luminesced pretty low when all the cells should have been live and luminesced higher. Also, the cells that were not treated with any compounds and only STS were pretty close to my positive control. So we should have seen a greater difference between these two. And also BDNF, another one of my controls. Also, the error bars, you can't see them too clearly here, but they're all pretty much overlapping, which you don't really want in experiments. You want to be able to tell and see a huge difference between a positive and negative control. And so my other compounds, I did a concentration titration and didn't really see a trend. So from 500 to 50, you should have seen an increase or decrease in luminescence. And with my other compound of interest, you didn't see a trend with these either. The only one that you did see a trend with was this one, although it's not a very drastic trend. When it's going to 50 to 6, or excuse me, 50 to 6.2, you should have seen a stronger trend. So this says our assay is extremely sensitive, or I need to narrow in the parameters. Maybe the age of my cells were too young, or other parameters that I need to tweak in order to get a more accurate reading. And so next, I wanted to develop an assay that, again, looked at this neuroprotective property, but more specifically, I wanted to look at the cells, the nuclei of these neurons. So what I did a DAPI staining. I grew cells on cover slips, then I fixed them, then I added DAPI to the cells to luminesce my nuclei. And so just a couple of examples of what I was looking for. So you see here, these are the nuclei of my neurons. These are the healthy ones that I counted. And these are the apoptotic neurons that I counted. So what I did is I took the damaged over the total, and this is how I created my percentages. So again, I'm seeing that the cells, the negative control and the positive control lied pretty closely. The error bars were overlapping. Although I did see some interesting neuroprotective properties for some of the compounds. So although these lie pretty close within the range, these did show neuroprotective properties because, as I said, it's damage over total, so the lower the number, the better, the more protected these cells were. So, in conclusion, I wanted to develop these assays that pretty much would be able to test the efficacies of my candidates of interest. So, initially, I looked for an antibody that would stain properly my phospho-TRK and found that my antibody did not bind or label specifically. So either our lab's going to have to invest in making an antibody, or ordering one, or trying another site, because our site was tyrosine 186, so maybe a different tyrosine. Also, I developed an assay or I attempted to develop an assay to look at the cell viability, the neuroprotective properties. So I found that my cell titer glow was either too sensitive or the assay didn't work well at all, because our positive control and our negative control lie too close to be able to differentiate and test my candidate molecules. So either I throw it out or I find another one. And also the nuclei staining, the DAPE staining that I did for the damaged neurons. And what I found that, again, the positive control and the negative control lie within close range of each other, although I did find some interesting neuroprotective properties. So I could increase my sample size to find more robust results, or I need to treat them longer with my STS or my flavonoids of interest to get a better understanding of the damage to the nuclei. So I would like to thank Amgen for funding my research this summer and giving me this opportunity, and my SSRP, everyone here, specifically Terrence, Summer, and Trisha. Trisha helped me make this slide every single week and just helped me improve it and improve myself. My PA group that listened to this a million times, for better or for worse. All of you guys for listening. My lab, which showed me a complete different side of science. Jay for giving me this opportunity in this lab. Don and Jay for answering all my silly questions this summer. So, thank you.